Hello students, in today's class we will see how the genome data that we retrieve or use for our analysis, how it is organized in various databases, what actually is a database. So we will have an overview of the commonly used science databases that are generally used for genomics. Okay, so let's proceed. Basically, first of all, let's understand what is a database. So these days we are generating information from all over the world. People are doing a lot of experiments, generating sequence data, proteomics data, metabolomics data. So a lot of data is coming from various parts of the world, from various laboratories. So it is required that this data is organized at some common or publicly available places, so that the other users can use the data when they need, and they know that what is going on in the other parts of the world, so that when you are also doing something similar or that you need some data for your analysis, you can retrieve from there. So that is why data storage and organization is very, very important. Okay, so that is why we have several database management systems, and data science is also emerging as a very important area. So what is a database? So basically, database is a structured collection of data. Okay, so we cannot randomly store the data, because it could be multifaceted, it could be involving multiple aspects. So we have to properly annotate it, save it, retrieve it. For example, when you search Google, there is so much information that for one query you can get so many hits. So all that data is organized in such a way that you can query it, you can retrieve it, and also at the back end you can store it and save it. So data is basically structured in a database, which is easy to access. You can make queries, and also databases can be connected to other databases. So database management and structure and organization is very, very important. And at the same time, data security, safety, curation, and somebody has to check that whether what you are submitting or what is available over there is correct or not. So all these are very important aspects of data. So a database refers to data, and they are supporting data structure. So we will see today that there are some database management systems which are often used for this. Some are freeware, some are have some subscription fee. So okay, so these database management systems makes it possible to organize the data in such a way that you can retrieve it very fast, and you can also make some structured query. Basically that language is called structured query language, SQL, so by which you can retrieve the data. Okay, so commonly used database management programs include like Oracle, FoxPro, Microsoft Access, Microsoft SQL, MySQL, which is available with Linux, which is commonly used. Okay, so different query languages have basically like some are available with like freeware such as Linux. Okay, and some come with some subscriptions such as Microsoft Access or Oracle, but they have their own capability to handle how much data you can save and retrieve at one point of time. Okay, so basically when you see data structure, it has to be organized, and also you have to retrieve, so for which you require algorithms, right? And okay, so basically, and one common word that you see these days is data science. Okay, so in data science what you do is basically the data is there all over the world. Okay, so you are extracting the data and also making queries and doing analysis from that data. Okay, you can do data analytics, you can create a new curated data from the available data. Okay, and also make predictions from that data. Okay, so this is very important area today, which is called data science, which helps you to organize, process, and use the data for various analysis and predictions. So as I mentioned, there are several advantages of storing the data at a common place and using a database management system. For example, if you are storing at a common place, so for example, NCBI is one of the resource. So all over the world, the scientists save their data or upload their data at NCBI, okay, which checks what you have submitted, and then it releases for users. So you can download the data from there, okay. And it is structured and standardized because it is structured so that you can see that which type of data you want to download, and also available in a standard format, so that you don't have to worry about how the data was submitted, right. So the user is also submitting in a standard format, and you are also retrieving that in a standard format, so that you can do analysis and use your scripts on that available format. Data from dissimilar sources can also be interconnected and used together, okay. So sometimes the data is available from multiple platforms, multiple technologies or system that has generated the data, okay. So all that data has to be brought to a common platform, okay. So this is another important requirement of a database management system. It should be verified so that you don't have to worry about the quality. So NCBI also does these kind of checks that the data that is submitted, like it could be sequence or protein, it is routinely verified and organized, okay, and available to you in a standard manner, which I have mentioned. There has to be easy and rapid access of data, so that you don't have to wait to download something, okay. So as soon as you click, you are able to download. And if the data is too big, okay, at your end when you want to upload or to download also, so FTP links are also provided. So we will see what is FTP in the later part of our talks, okay. So it provides a platform by which you can upload a lot of data, large amounts of data, and also retrieve those data by using a username and password, okay. And when you are downloading, there are several other people are also downloading the same data, okay. So basically this data is available for multiple users at the same time. So this is another advantage of using a common data and database management system, okay. And so you are downloading the data for your purpose, somebody else is doing for their purpose. So data can be used directly in various programs or applications that you are developing, okay. And so basically a database management system such as MySQL or Oracle or FoxPro or Microsoft Access provide you various capabilities to organize that data, okay. For example, you can create a new file, add something to the existing records, delete a file or a particular record, or modify a particular information, okay. Retrieving the data collectively or selectively, that means you want to download the entire database or a part of that, okay, while at the same time maintaining the data integrity and the accuracy, okay. Data can be sorted or indexed the way you want. So you can define your own private and public keys or foreign keys, okay, by which you can access the data or interconnect the data from multiple sources, okay. It also reports errors and anything that is a warning or regarding the data. You can perform mathematical operations on the data so that many times when you get the data, okay, you want to see that, okay, basically data is what? The data is composed of multiple files. For example, one person made a file from, say, organism name, its accession number, and its source, okay. Another person made a file for the same organism, like who published that data and who authored that, okay. So basically the two tables will have something in common. You can connect those two tables by using the common keywords, okay, and then you can ask or query that, okay, what information you want from multiple tables like that together, and download that information. So when we are organizing and storing the data, we also have to worry about the security and safety, because editing one record and one information can lead to major errors, right. So basically data is secured using passwords, and various levels of access control are there in the database management system, okay. And there are query languages which are available to query the data, okay, as I mentioned previously. So one is structured query language which can be used to query all the databases. So mostly all the query languages are similar in syntax, right. So SQL is one open source freely available query language, which is also available with MySQL. Okay, it is simple to learn and basically easy to operate, and okay, you can learn it easily. Okay, so basically this SQL learning is very important when you want to access the data, you want to save the data, retrieve the data, or make any changes, add something to the data. Okay, so now with this brief introduction of what the databases mean, we will show that what are the commonly available genomics databases which you can use for genomics analysis, proteomics analysis, and many times we require data from multiple of these sources together, while doing some analysis. So the analysis is also subjective, depends upon what question you are asking. So that is why there are so many variants of databases and data is available. Okay, so so now when you see data common, like broadly we can classify into two types, like the nucleotide data or the protein data. Okay, so some commonly available nucleotide databases include say EMBL, DDBJ, NCBI. NCBI is one of the most important, where you can access almost all kinds of data in the raw form and semi-curated form also. UCSC is one. Okay, then for protein, again NCBI is one of the important source, and then PDB, which provides the structures of proteins and coordinate and those X-ray crystallography data also. SwissProt, Ensembl, and for pathways, that is both protein and nucleotides, you can have KEGG database. Okay, so NCBI is one of the oldest and a standard database that is used by researchers worldwide in the area of genomics, proteomics. Not only data, it also provides you several tools. It houses the PubMed database, which is the biological literature. Whatever papers it publishes are available at PubMed, so you can use that information, right? So it is National Center for Biotechnology Information. So basically they organize and run this database. It is publicly available at www.ncbi.nlm.nis.gov. So this is the say front page of this database. So I will show you how this looks like. So if you see, so now we are at the NCBI database site. Okay, so if you see here, you can do login. Okay, so you can create your own login. You can register with NCBI database. This will help you to when you want to retrieve the data, right? So you can use their services for retrieval of data. Okay, so you can create a login for yourself. Then it provides the option to submit, download, okay, and also provides you a list of what all services and data that they provide. So now if you see here, so all databases, so if I click over there here, it shows you all the possible databases in which the data is organized and is available for download. So I can make a query, a general query in the Entree system. So this search system is called Entree, NCBI Entree. So I can say a query, for example, protein kinase. Okay, so it's a protein. So if I search in NCBI, so it gave me the information from all possible databases that NCBI harbors, that how many hits you can get for that protein. For example, in PubMed Central, you can get almost 1.3 million hits in gene, in proteins, in assemblies, in genomes, right? You can get several hits. Like there are more than 3 million proteins which are available with the keyword protein kinase. So basically it will not only search for protein kinase but also search for anything that is related to protein kinase. Okay, so that keyword has to be there in the the query line, or for example protein kinase associated protein. So that will also come when you search protein kinase. Okay, so you can click on any of these and you can get more detailed information about it. Okay, similarly I can search for example I go to PubMed. Okay, so PubMed is the database of all the journals which are indexed, okay, and available for users. Okay, so you can search what all literature is available for a particular keyword or a topic. Okay, so again if I search for example protein kinase here, right, so it gave me the hits where this keyword was available, like protein kinase classification, right, and okay, so in total the article that we saw earlier, it there are the number of queries are like it is organized in multiple pages, right, and if I want to now read this in more detail, I'll click on this, so it will show me the abstract and other information about this paper. Okay, and the similar articles, how many citations, title, author, abstract, okay, which are all important for this. So this is a detailed format. Now I can save this information also, okay, in the form of a text, okay, or just the PM ID, which is the PubMed ID, or the abstract as a text. For example like this, if I say, so it will create a file which I will store locally. Okay, so this will go as a file in your system. I am not saving right now, but you can do the same. Similarly, you can email to yourself, or you can send this information to a clipboard or basically to a citation manager, because you have to quote this information later or cite this information in your work. So this will, the PM ID will be saved for later use. Okay, now I can also do an advanced search here. So here what I can do is I can say that okay, the field that or the query that I want to search should appear in the abstract, okay, say title or abstract, right, and search term can again be protein kinase. Okay, now I can also use the 3 Boolean operators, which are and, or. Okay, so if I say like and, okay, so it will search for example protein kinase and, okay, protein kinase and, so I will say that human. So it will search protein kinase associated with human. Okay, if I do search now, so it will give me hits where both the words protein kinase and human are appearing. Okay, similarly I can use another operator, Boolean operator, or here. So either it should contain protein kinase in the title or abstract which I have defined. I can also pick the entire journal, or I can also search by author. Okay, and various other information which is available in PubMed. So if I search like protein kinase or human, so it will give me wherever the 2 terms, any of those, protein kinase or human, are appearing. Okay, so that will be like too much, because human is a very common word and you have to use the words very carefully. Okay, otherwise in human is one of the words that may appear in like millions of articles in NCBI. Okay, so for example here also it gave you like 3.1 million hits. Okay, similarly I can say that in place of or I can say not. So basically and, or, not are the 3 Boolean operators. And means A and B both should occur in that search term or search area. Okay, like in the title of the paper or in the abstract. Okay, both the 2 terms that you are defining should appear. Okay, similarly or, so A or B, either A or B should be present in the area where you are searching. Okay, and now if I say not A not B, That means A should exist but B should not exist. So if I say protein kinase not human, so it will give me information on protein kinases, but in that abstract or title of the paper, the human will not appear. Okay, so basically protein kinase not human. Okay, so it could be mouse, it could be like other species, but human will not be there. Okay, so this way you can make the Boolean operators, and not only this, you can also extend it further. So it is using the same syntax, I can add one more term and okay, maybe define multiple like A and B, okay, or C, A, okay. So basically B or C you can put in the same bracket, okay. So that way A will be present and B or C, any of those will be present, B or C, okay. Similarly, like whatever you have searched or in NCBI, it appears as a history, right? So which is available here, so that you don't have to remember that what type of queries or searches you have made. Sometimes you are refining your query, but you can again go back to the previous query from this history. Okay, so now again coming back to NCBI. Similarly, there are options to submit your data and retrieve the data. So we will see this part a little later. Okay. Okay. Now if I say nucleotide, okay, so in genomics we need the DNA RNA data, right? So that is housed in the nucleotide thing. So again, if I say protein kinase, so it will give me all the gene sequences where, or the sequences where the word protein kinase will appear. For example, the entire chromosomes will come, and also at this the particular protein kinase, if I make it better, a more refined query like Homo sapiens receptor tyros, okay, so something that I choose from the hints that it showed. So you can see that it will give you the searches where the word Homo sapiens, which are highlighted in dark blue, and the other keyword like receptor tyrosine protein kinase is appearing, okay, and right. So and on the right hand side you can see that it also gives you a hint like from which species your queries are appearing, like Homo sapiens or other other taxa which are available here. So again you can click here and directly go to what is the source of that information, that like for example protein kinase. Okay, now again if I click on these, I can, like for example these two papers are of interest to me. Okay, so basically you can select multiple of these also or all of these also, and you can specify that what should be saved at your local directory. So I can say that okay, all the FASTA sequences or FASTA sequences in text. Okay, so now it will show me the FASTA sequences of these proteins. Okay, so these are the gene sequences for these two proteins, right, in the FASTA format. So now I can say that okay, select these, both of these, and save it from here, right, to my, and send to my as a file, okay, in FASTA format, default order, or you can also specify the order in which you want to save, like based on some criteria that shorter sequence should be first, or taxonomic ID, or date of their submission or release, okay, and then create a file, so it will be saved locally in your system, right. And similarly for PubMed that we saw, or protein, okay, or any other information that we are using from NCBI, we can make Boolean queries using the Boolean operators, and also save the results in our local system, okay, and a history will also be saved for you by NCBI. So if you log in, it will be better, because then you can have your own login for or basically access to NCBI personal page, and you can see that what all you did, your activities there. Okay, so the good thing about NCBI is that it also gives you some of the tools for analysis, right. Although the tools are basic, for example ORF finders, or finding some pattern, or checking your primers, there are specialized databases and softwares for doing the same job, okay, but still those options are available, right? So NCBI is actually collecting data from all over the world. So whenever say I have to publish a paper where I am telling about a new genome that we have sequenced, or a new gene or a marker, so we have to submit that data to NCBI. Okay, so there is a format to submit the data, and in the format we have to enter like the information that is desired by the users or by NCBI, so that other people know that what is available and how it is submitted. Okay, so with that okay, so you submit that data, NCBI will check that data, and then they will release that data for public viewing. So basically for all the data, they or if there are multiple sequences, so accession numbers will be provided to those sequences, okay, which are the you can say unique identifiers of that particular sequence, right? And it is mandatory that when you are publishing a paper, you have to submit the data to NCBI, okay, so that later on people can retrieve the data, and this way data sharing is possible, and validation also happens indirectly, because your data is now open to public and they can use that for their own analysis, and whatever like question they want to ask from the same data. Okay, then NCBI is one place where you can get like the raw data or slightly polished data for various organized into various sub databases. Now if you want to know that how many projects are running all over the world, and okay, so basically JGI or Joint Genome Institute Gold Database, Genome Online Database is very important site for that purpose. So here it gives you a snapshot of, for example, metagenomic data sets studies which are available are 4,000, okay, non-metagenomic are 54,000, and then host associated biosamples. Okay, so basically when you submit a data, human is a sample, so it's a biosample, and similarly it gets an accession, right, later. So it gives you information about host associated, engineered or environmental. Complete sequencing projects are 34,000 all over the world. Okay, there are more than like 3 lakh draft genomes available. Incomplete projects which are still going on are 164, and this is updated like often, so that you are updated with what is going on. And if you click here, you will know that the list also of all the completed and ongoing projects. For example, metagenomic analysis projects are 1 lakh, and okay, so basically here organisms which are sequenced so far are 4,88,000, archaea 5,000, bacteria more than 4 lakh, eukaryote 47,000, virus 18,000. Okay, so it gives you a snapshot of all the genomes which are available for access. Okay, and when we click here, for example, if I click on bacteria, it will give me a list of the bacterial genomes which are available with a unique GeoID. Okay, and if I go to, for example, this is for say one genome, Photobacterium rosenbergii. So if I click on this, it will give me more details about who submitted that, where it is available, and how you can download the data. Okay, so this is a one place where you can know that what all projects are going on all over the world, and also it is connected to the other databases where the data for that particular project might be saved. Okay, similarly there is another website or database called EMBL EBI. Okay, European Bioinformatics Institute is running this. So this also provides you a kind of curated or polished data. Okay, for example, the raw data is submitted to NCBI, but that data, another group of researchers or such as EBI now takes that data. Okay, they may also generate their own data, EBI, or collect from other places, and then they curate or polish or fine tune that data and give you a high quality data for the, for example, you can search a genome mouse. So the raw data that was submitted to NCBI is available at NCBI, but the polished genome sequences, sometimes it could be alignment, sometimes it could be the complete contiguous sequences or assembled sequences may be available at EBI. Okay, so this way EBI is an important site. Okay, then there is another DDBJ, DNA database, data bank of Japan, which is another source of the information about the genomics data. Although this EBI, DDBJ, NCBI are all connected, and they also talk to each other that about what has been submitted to their databases and also keep themselves updated. Okay, so these are some of the commonly available genomics databases, although NCBI and these also give you a lot of protein information. Okay, but the dedicated protein database is SwissProt or Expasy. So this database provides you basically, it is a house of protein sequences and enzymes, right? Which are classified into 6 EC categories, enzyme commission categories. Okay, so it gives you information about the curated proteins as well as also the predicted proteins. Okay, so it has UniProt and Ensembl, 2 databases, right? So UniProt is a expertly curated component of UniProt KB. It contains thousands of protein descriptions, function, domain structure, post-translation modification. So basically, if you see here, the protein data that was submitted to NCBI is not available in a curated or polished form. Curation means that somebody has looked into that sequence which was available at NCBI or as a raw data, and checked that sequence, verified, and then it is available now at SwissProt. Okay, so when you do your analysis, if you use the SwissProt data, basically you are using the data which is validated or verified by somebody. Okay, similarly it also has the Ensembl or the so basically if you see here, there are so many other things that you can do here, in terms of the way they have organized the protein information or data sets, right? And you can click and go to those, right? And the other part of this is the Ensembl, which contains the data which is the protein sequences which are computer verified and computer generated also many times, predicted protein sequences, you can say, right? So then proteins do also have structures, right? Because in 3 dimensions, so which is done by X-ray crystallography, and those data in the form of the structures and coordinates are saved at the PDB database. Okay, so this is also updated very frequently, and you can download the information about for what protein the structures are available and their at what resolution and their coordinates also. Okay, now I will show you one more database which is called UCSC Genome Browser. So it harbors a lot of data, also provides you some tools like BLAT. So BLAST is one tool for alignment, and BLAT is a little faster than BLAT. Okay, so we will deal with these tools later, but today we are discussing about genomes and their analysis. So Genome Browser is one of the important component of UCSC database. So like if you see here, it's a Genome Browser database. So it is asking you like where to take you, the local Genome Asia or the main Genome UCSC.edu website. So basically, since we are from Asia, I can click here. So it will take me the mirror website which is hosted in Asia. So now here it is giving me information about like human assembly, which is December 2013. Okay, so I can click. Okay, so basically this is December 2013 HG38 assembly of human. So and you can also see that there are some other assemblies also available. For example, you started your research maybe 20 years back. Okay, so you might be dealing with HG16 human assembly, but after that new changes, modifications, additions to human genome might have happened. Okay, and that is why 16, 18, 19 and now like HG38 is available. So Okay, so you would like to stick to that assembly for your analysis. Okay, then you have to provide some search terms. For example, chromosome 2, you want to find something on chromosome 2, okay, right, which is starting the from the locus on the chromosome is this 251609115 to 251689903. Okay, so this is the location on the chromosome 2 that you want to see. So it will take me to that location. Okay, so now this is your region in the chromosome that I entered. So chromosome 2, and from 25 million to 915 to 25 million. So basically about 8000 base pair it is showing me here. Okay, so I can zoom in or zoom out. So I can say okay, 10x zoom in. Okay, so now from 8000 it has moved to 800 base pair, and I can see the region in more detail. So similarly, if I zoom in even further, okay, so zoom in say at the base level, I can also see the ATGC level sequence of the chromosome like here. But since we have gone so much in deeper in the chromosome, it is only the nucleotide level, but we have missed out on the structure of the chromosome and what genes and what things are available. So I can say like now zoom out, say 100x. So now we are coming back to like, now it is showing me 15,000 base pairs. Okay, now you can see that this gene is complete here. Okay, these are the 3 exons for this gene. Okay, even I can click on this exon and get the information about that exon sequence, right? And you can see that it is organized in the form of tracks, okay, which are many, which I can switch on and switch off using here. Like I can say switch off, I can hide this track, okay, and say refresh. Okay, so it will hide the track which I have just removed. Okay, or I can add the tracks. So basically you can play with this, and it has a lot of information about what you want to show there, like gene sequences, okay, encode tracks. Okay, maybe I can hide all of these. Okay, repeats, I can hide this also, and just keep the genes and just say refresh. So now it will remove those tracks and give me like lesser information. Similarly, I can add more tracks. So UCSC genome browser is a good place which can be used for comparative genomics, and for zooming into a region of chromosomes for a particular assembly that you are interested in, and to view what genes are there in that region, their expression patterns, their conservation across genomes. Okay, so UCSC genome browser is a site for that. And okay, so basically spend some time on this, and I think this will be useful for you. Okay, and then one important thing that we usually need in our analysis is the metabolic pathways. Okay, because genes do functions in various pathways. So genes are expressed as RNA and then protein, and then proteins are part of a particular pathway where they act on a substrate and convert that into a product. Okay, so all that information is organized in the KEGG pathway database. Okay, this is a database of Kyoto Encyclopedia of Genes and Genomes, KEGG database. It is hosted by a Japanese group. So for example, if I say a carbohydrate pathway, so it will like glycolysis, TCA, and all the carbohydrate metabolism pathways available here. If I click on say glycolysis, it is taking me to glycolysis pathway. Okay, where you can see that these are the various enzymes, right, and how they are connected to each other. So product of this goes to this enzyme and converts into that. So I clicked on one particular KOID, and it is showing me the detailed information about that KOID 1623. Okay, in which all pathways it is involved, because many a times one protein might be involved in multiple pathways, right? So this is gives you information about, okay, that also. Okay, so KEGG is another important pathway that basically utility that gives you information about the pathways, and you can use this for various pathway analysis. In my slides I will now show you a little bit of this. So NCBI we have discussed, so it's a database of all the it will refresh. So what I am now telling is going to refresh what we have done, and also this will also be available as a notes to you. Okay, so NCBI is the common database that houses all the genomic, proteomic and protein data, and PDB and links. And so basically the entire world's data is kind of saved at NCBI, and you can use that in addition to the literature information. Okay, so this is the NCBI site, right? So NCBI also have a sequence read archive, okay, which means that when you are storing the data or basically submitting your data to NCBI, it goes to a archive. Okay, so from several years they are maintaining the data, so they have the various versions of the data also. You can go back to previous version or the current version. Okay, and you have to submit your data to NCBI SRA when you are doing any research or submitting a paper. Okay, then we discuss about the Boolean operators, which are and, or, and not, which you can use to query the NCBI database, right? So these are the three Boolean operators, and, or, and not. All have to be put in the uppercase, because and, or not can also occur as a like common word in any title or abstract. So they have to be in uppercase when you are making a query, right? So like this is one search result using, for example, I searched an author called Vineet K. Sharma, and it gave me information about all the papers the authors submitted. Now I can use the another Boolean operator like Vineet K. Sharma and protein, so it will search all the papers where some research on protein has been done by the same author. Okay, then this is a list of the protein databases in which we have today discussed Swissprot, Tremble, and there are so many other databases if you search Google, because people are developing these databases as a part of their research, and they are the ready source of curated and you can say knowledge which you can use for your research. Okay, then there is a term called non-redundant database. Okay, so for example, there is a so basically this non-redundant database you can create on your own, and this is also available at NCBI for download, okay, with the BLAST tool. So non-redundant means something which is not repeated again, okay, or unique. Okay, so for example, there is a protein, okay, which is composed of say amino acids. So the exact chain of amino acid is present in several genomes, like as is identicals, and you will expect that the genomes which are very close to each other, okay, showing like identity at the strain level or the species level, sometime have the exact sequence of those amino acids. Okay, so if I put that sequence in the database, so like genome A with the sequence and then genome B with the sequence, so basically I am repeating the same sequence which is commonly present across multiple genomes together. So instead of that, what I can do is I can use that information and okay, make it unique, so that only one time I will repeat that sequence and add in the annotation line A, B, C. Okay, that means the common sequence is present in like as a identical sequence in genome A, B and C, and like that. So non-redundant databases, we will also see that in detail when we go further. Okay, Swissprot is the database of proteins that we discussed today. Okay, this is the Expasy site. Okay, this is the protein data bank site which we have discussed today, right? So basically it's the source of the structural coordinates and who solved the structure, and because these structures are needed for molecular dynamic simulations and finding some active sites, or even improving the structure further, or comparative modeling also. Okay, Then we also see today EMBL EBI site. Okay, JGI Gold is a very important site which gives you a summary of what all is available worldwide, what projects are going on in what category, and you can access that information. Okay, and we also saw JGI Genome browser, which helps you in the comparative analysis of various genomes and genomic sequences, right? Okay, so we saw an example of that today. And we also saw KEGG pathway database, which is a prominent database for all the pathway related information. Okay, so it gives you information about what genes and proteins are involved in a pathway, in which genomes they are conserved, how the pathways, which pathways are present in what genomes, right? And you can also map this in on your genomes also. For example, this was one metabolic pathway. Okay, so you can say carbon metabolism. This is say the 10 steps of glycolysis and the TCA cycle. So you can see that, okay, this is everything is highlighted. This is a reference metabolic pathway, and when I do for a particular genome, I can see that, okay, the genes which are present in that genome will be highlighted here. Okay, and the remaining parts of the reference pathway which are not present in a genome will not be marked. So this way you can know readily that what information is present in that genome and what pathways are present and what are absent. Okay. Another important pathway is metabolic database, because metabolomics is becoming a very prominent field these days, because ultimately what is made, or the final product of all the metabolic pathways, we would like to see. Okay, so this houses information about the products of various pathways, their reactions, and the resulting metabolites also. Okay, so another database is HMDB metabolomic database, which is houses about more than 2 lakh metabolites from various enzymes and substrates. So in this class we covered how the genomic data is organized in public databases and their structure, and the various genome web resources which are available, which provide this genomic data, including the nucleic acid, DNA, RNA, protein data, sequence data, or the expression data, right? And also we have also looked at the various metabolic and the metabolomic databases, which are very important for the finer analysis, and metabolomic particularly is also becoming very important area these days, because there we can measure the output or the final product of a metabolic reaction. So thank you for today, and see you in the next class.